Hello. Okay, you can hear me all right? My name is Mathieu Virazels. I'm a second-year PhD student. My project is supervised by Bruno Segui, you already know, and Céline Colacio. And today I will talk about the systemic immune responses that we observed in patients, in melanoma patients receiving TNF blocking antibodies combined with immune checkpoint inhibitors. So cutaneous melanoma is the deadliest type of skin cancer, and it originates from melanocytes, which is the pigment-producing cells of the skin. And when taken care of at early stages, melanoma is of good prognosis because most of the time surgical resection is sufficient. But at later stages, melanoma has a poor prognosis. So in the last decade, the treatment of metastatic or advanced melanoma has been revolutionized by immunotherapy, and with the use of anti-PD-1 and anti-CTLA-4 antibodies and the combination of the two, we can now achieve nearly a progression-free survival of nearly 40% of patients. So this is a major breakthrough, but this also means that more than half patients do not respond initially or eventually relapse. On top of that, immunotherapy or immune checkpoint inhibitors are associated with immune-related adverse events that can affect various and numerous organs, and they are experienced by nearly all patients receiving the combination of anti-PD-1 and anti-CTLA-4. And 60% of these patients develop grade 3 or 4 immune-related adverse events that lead to treatment discontinuation. And these events are managed with corticosteroids or infliximab when they are refractory to steroids. And infliximab is an anti-TNF antibody that is a powerful immunomodulator. And whether TNF blockades affect the outcome of immunotherapy is still unknown. So I won't get into too much detail here, but the role of TNF in cancer immunology is really ambivalent, because on one hand, TNF has some beneficial effect on the anti-cancer immune response, and on the other hand, it has some immunosuppressive effects. So we postulate that the immunosuppressive effects outweigh the beneficial ones. And this is because in preclinical models in murine melanoma, our team showed that blocking TNF improved the efficacy of anti-PD-1 therapy. And this was associated with an increase of cytotoxic T cells infiltration in the tumor and a reduced activation-induced cell death of these infiltrating T cells. Another group, the team of Ignacio Melero, showed that blocking TNF can improve the efficacy of the combination anti-PD-1 anti-CTLA-4 in murine melanoma and colon cancer. And it could also reduce the toxicity of such a combination, especially on colitis and hepatitis. So based on this preclinical data, a phase 1b clinical trial was designed and is conducted here in Toulouse. It's called TCML, and the PI is Nicolas Meyer. Bruno is responsible of the exploratory part. And all the experiments I will show you today were performed by Anne Montfort and by myself. So in TCML, patients received the concomitant combination of anti-PD-1, anti-CTLA-4, and anti-TNF antibodies. There are two groups, one receiving nivolumab, ipilimumab, and certolizumab, the other receiving nivolumab, ipilimumab, and infliximab, so two different anti-TNF. So Bruno already showed you the clinical results, and the goal of the exploratory part that I will show you now was to evaluate how TNF blockade affects the systemic immune response to immune checkpoint inhibitors. So which cells do we study? We mainly focus on T cells because, well, it's the target of anti-PD-1 and anti-CTLA-4 antibodies. And it is well known that those are the the key players, the key cells that are responsible for the efficacy of immunotherapies. So under anti-PD-1 and anti-CTLA-4 treatment, cytotoxic T cells and helper T cells proliferate and reinvigorate. It means that they regain effector functions. And that is crucial because, as you may know, cytotoxic T cells are the main tumor killing force. In helper T cells, and especially type 1 helper T cells, there is also proliferation and a secretion of interferon gamma, which is the main anti-tumor cytokine. And on the other end, some cells are detrimental to the anti-tumor immune response, such as myeloid-derived suppressor cells and regulatory T cells, two types of immunosuppressive cells that can inhibit the anti-tumor immune response. So this is our workflow. So before and along treatment, we receive blood samples. On those, we perform flow cytometry analysis. We also separate the plasma to evaluate the plasma cytokines levels. And we also isolate PBMC, so peripheral blood mononuclear cells, to perform further flow cytometry analysis. So first, circulating T cells. So you have on the left patients receiving ipilimumab, nivolumab, and certolizumab. In the middle, patients receiving ipilimumab, nivolumab, and infliximab. And on the right, all patients receiving ipilimumab, nivolumab, and either certolizumab or infliximab. In blue dots, you have patients that responded to the therapy, and in red dots, patients that did not respond. So you can see that after 6 weeks of treatment compared to baseline, we observed an increase of the total T cell count, as well as an increase of the CD4 helper T cell numbers and CD8 cytotoxic T cells. This was associated with an increase of the expression of Ki67, which is a marker of proliferation, and in T cells, proliferation is a sign of activation. So for both therapies, we saw increased peripheral T cell numbers and proliferations, signs of activation, of peripheral activation. So now a word on T cell differentiation, because to have a proper and effective immune response, T cell must be educated and gain effector functions. So we studied 4 main subsets of T cells: naive, central memory, effector memory, and terminally differentiated effector memory cells. So for the naive compartment, you can see that we did not observe any significant changes at week 6 compared to baseline. For CD4 cells, CD4 helper T cells, we noticed an increase of central memory and effector memory cells. And for cytotoxic T cells, CD8 T cells, we observed an increase of effector memory and terminally differentiated effector memory cells. And this for both cohorts. So for both cohorts, we saw a differentiation of peripheral T cells, which means that these cells have encountered a target antigen and have been activated. Now a focus on type 1 helper T cells. So this is a subpopulation of helper T cells that is known to be antitumoral. And for these cells, we saw an increase during treatment compared with baseline in the number of these cells as well as in their proportions. And this was associated with an increase of plasma interferon gamma levels, which is, as I said, the main anti-tumor cytokine, and it's produced mainly by Th1 cells, type 1 helper T cells. So for both three therapy and actually all patients, we saw an increase in peripheral Th1 cells and also cytokine plasma interferon gamma. Beyond that, we saw no significant change in the proportion of Th2 or Th17 T cells, which are two other subsets, two other subpopulation of T helper cells. So now a quick word on myeloid cells, and specifically monocytic myeloid-derived suppressor cells, because these cells are immunosuppressive and they can inhibit T cell activity. And during treatment, we saw that at week 6, a decreased proportion of these cells in both cohorts. And this is a good thing, considering they are immunosuppressive cells and inhibit T cell response. So to conclude and to summarize what I showed you, patients in TCML received a concomitant combination of anti-PD-1, anti-CTLA-4, and anti-TNF antibodies. And during treatment, we observed proliferation and differentiation of cytotoxic as well as helper T cells. We observed a Th1 polarization of helper T cells and an increase of interferon gamma plasma levels. And finally, we observed decreased proportion of monocytic myeloid-derived suppressor cells. And all these signs are, all these observations are signs of an active and promising peripheral immune response, promising activation of the peripheral immune response. And most of all, the combination of anti-TNF blockade with anti-PD-1 and anti-CTLA-4 seems to be activating the peripheral anti-tumor response and not shutting it down. So you may have noticed that all the changes we saw at during treatment at week 6 were not sufficient to discriminate patients responding to those who did not respond. So how to predict and to explain the response of patients? So to do that, we are performing transcriptomic analysis on the PBMCs of the very same patients. And the analysis of the site-seq and bulk RNA-seq are done in collaboration with the technology cluster at CRCT and the team of Vera Pancaldi, and especially by Mathieu Virazels, who is a fellow PhD student in the team. And he will talk about this just after, I think. And I will let you guess who is Mathieu on the picture. So with that, I would like to thank the organizing committee for giving me the opportunity to present my work for the first time today. Thank you. Also, I would like to thank all the team and especially people that are working on this project, all our collaborators and our funding. Thank you for your attention, and you're welcome to ask me questions.